On behalf of myself, my fellow co-chair, Michael Nagel, and our FNIH project manager, Francesca Signorella, I want to welcome you to the AMP80 session. It's a real honor and a treat to be here today and tomorrow to celebrate our collective achievements and to learn from each other. So, exactly 10 years ago, we launched the Accelerated Medicines Partnership for Alzheimer's disease. And we set out to do two challenging things: find a better way to qualify existing targets, and then find new ways to identify better targets for this complex disease. So we did that through two distinct projects. The Biomarkers in Clinical Trials project, where through the partnership we were able to embed the emerging tau PET ligands in two secondary prevention anti-amyloid trials. And the Target Discovery and Preclinical Validation project, where we harnessed the power of big data and jumped on the riding the multi-omics revolution wave and applied a systems biology approach to discover and validate the next generation of therapeutic targets. These two projects are seemingly quite distinct, but they have one thing in common. Both are marked by rapid and broad sharing of data and insights and open science practices. The NIA wanted to raise the bar on data sharing and open science, and we are very grateful that our industry and nonprofit partners embraced this and really pushed to fully realize this across the two projects. You will hear from Lori Ryan about the Biomarkers in Clinical Trials project, but a couple of words on the Target Discovery and Preclinical Validation project. And as you heard from Francis, many of the AMP projects are tackling the difficult task of deconstructing the complexity of these major chronic disorders. We started to do this in the AMP Target Discovery project, leaning on human biosamples and rooting this discovery in human data. This was designed as a large-scale team science effort, bringing together 6 multi-institutional academic teams and a data coordinating team at Sage Bionetworks. So there was a massive data generation effort, modeling endeavor, and early validation across multiple experimental systems. This was only made possible and glued together through the development of a centralized data infrastructure and the sharing of all aspects, all deliverables, from raw data through target insights. So in the first 5 years, the Target Discovery project really delivered unprecedented amounts of multi-omic data, new mechanistic insights on the disease, and most importantly, perhaps, put in place a data-driven process for target discovery and early validation. We wanted to become more ambitious in the second phase, and together with our partners, conceived of a partnership that will take that pre-competitive open science enterprise, expand it to enable a true precision medicine approach to target and biomarker discovery. And given the complexity of the disease now, the outputs are not just open data, methods, mechanistic insights, but we're going after disease subtypes, and not just targets, but also biomarkers. So this was realized through a suite of foundational grants supported by the NIA, on top of which we were able to embed the multiple work streams that are bringing together massive amounts of data from across diverse donors. In particular, African American participants and Latino American participants. These are populations that are disproportionately affected with the disease and we wanted to make sure that we explore the molecular complexity of the disease across diverse cohorts. Again, so these components, all these components are being executed by an even larger cross-disciplinary team. These are 7 multi-institutional academic teams across 20 universities and the Data Coordinating Center team at Sage Bionetworks. Now, of course, we can spend a long time expressing gratitude to everybody who contributed to the success we've had to date. Wanted to acknowledge our fellow public sector partners, the National Institute of Neurological Disorders and Stroke, and the FDA, and our private sector partners who have supported us and contributed to the envisioning of this enterprise and its successful implementation over the last 10 years. There are also many people to thank, of course, the large human network of researchers who are executing on the vision set out for the partnership, all the members of the steering committees over these 10 years. On behalf of the NIA's AMP-AD leadership team, we are extremely grateful to our industry co-chairs, to the fabulous team at the Foundation for the NIH, and the project managers who made this partnership possible, and the team at Sage Bionetworks for really creating the glue and the platform for large-scale team science. So, over the next hour, you're going to hear from my fellow AMP-AD teamsters. Lori Ryan, Anna Greenwood, David Bennett, Nick Seifried, and Matthias Arnold are going to share our approach, our resources that we have generated to date, and the translational insights we've gained. And we're going to conclude the session with comments from Michael Nagel and Rebecca Edelmeyer, who are going to give us the public advocacy perspective and the industry perspective on the impact and the value of AMP-AD. Thank you. Laurie.
Great. Sorry, just have to adjust here for some of us of shorter stature. Okay, let's see if I can. Okay, hold on. These are not my slides. Hold on. There we go. Okay, there we go. Got it now. Thank you very much. And thank you, Susanna. It's hard to believe that it's really been 10 years of AMP that we've been working on this, and it's really been an amazing journey, and it's so great to see it continuing with all of the additional AMPs. So I'm going to talk about AMP 1.0, and specifically the clinical biomarkers and clinical trials project, as Susanna said. I'm going to adjust this a little bit because it's kind of strange. Let's try that. Okay, that's better. So as Susanna already mentioned, what this did was to enrich already existing NIH-supported secondary prevention trials using an anti-amyloid therapy, A4 and DIAN-TU, with adding tau PET imaging to test its utility really as a marker of progression and disease treatment. Okay, sorry. And of course, the mandate, which is really important because it's one of the first for clinical trials, was making this data, both the screening data, pre-randomization and post-randomization data, broadly available. And this really follows the CAP principles, which came out in 2016, which was a partnership among academia, the FDA, NIH, and also industry. So the screening and pre-randomization data had to be made available 12 months after the end of enrollment, and then post-randomization data and biospecimens made available as soon as possible, of course, without compromising trial integrity. And so the first of either after regulatory approval or trial completion termination or 18 months, whichever comes first. Okay, so I'm going to focus on my few minutes on the anti-amyloid treatment and asymptomatic AD trial, or A4. because we only have time to talk about one, and the screening data from A4 has really been particularly, I think, impactful. So it was the first of its kind secondary prevention trial in cognitively healthy older adults who had evidence of amyloid pathology on PET imaging. It was a randomized, double-blind, placebo-controlled phase 3 trial using solanezumab versus placebo for 240 weeks with an open label extension. The trial ran from 2014 to 2022, so it was a long trial, 8 years. There were 1,323 participants, but nearly 7,000 participants screened across 67 sites in the US, Canada, Japan, and Australia. There was an observational cohort of amyloid negative screened fails. That was the LEARN study that was actually funded by the Alzheimer's Association. The main trial itself, even without the tau imaging, was already a public-private partnership funded by NIA, Lilly, the Alzheimer's Association, GHR Foundation, and a number of other nonprofits. And of course, as I already mentioned, the data and biosamples met the CAP principles for sharing. Okay. So the trial top-line results came out in March of 2023, and the paper, the primary publication, came out in September of 2023. And unfortunately, it was a definitively negative study, but it was definitive. It was not a failed trial. So solanezumab did not slow cognitive decline in preclinical AD or reduce the risk of progression to symptomatic AD. There was not a slowing or stoppage of amyloid accumulation on amyloid PET. Amyloid continued to accumulate over time in both the placebo and treatment groups. Now, higher baseline amyloid levels were strongly associated with a greater risk of progression to symptomatic AD and a more rapid decline. And amyloid-related imaging abnormalities with edema or ARIA-E were uncommon and were similar between both the placebo and the treatment arms. Now, let's talk about some positive things. The data from A4. This is as of December 2023. This is the A4 screening data, and this is showing data, the data of the cognitive and clinical as well as the imaging data. There have been nearly 3,000 data requests, and this has been spread across many sectors, including obviously university and research institutes, biotech, pharmaceuticals, government, and also scanner manufacturers, and across the globe, really, but predominantly in the US and China, they're the largest users of the data. So this has been truly remarkable for a clinical trial, a pivotal clinical trial. This is the first one to actually use the CAP principles and have them executed. So I'm going to highlight 3 papers that have used the screening data. They're giving us really invaluable insights into the disease progression, how we might need to think about stratifying and looking at treatments for different groups. So the first one is a study by Casey Dieters, taking again, the A4 screening data, looking at amyloid PET imaging in self-identified non-Hispanic Black participants in A4. And so, as we know, a greater proportion of, unfortunately, the non-Hispanic Black participants did not pass the initial screening in A4 compared to non-Hispanic whites. This is mainly due to some of the cognitive measures, so that, of course, that does suggest some potential bias in race in the A4 PET data. Okay. Let's see. There we go. Okay. So this is showing the proportion of amyloid positivity across ApoE4 genotypes from the A4 screening data. The non-Hispanic Black participants had lower rates of amyloid positivity, as well as lowest continuous amyloid levels compared to non-Hispanic whites, overall and when matched for age, sex, and education. And the race effect on amyloid was actually strongest in the ApoE4 positives group, which might not be as surprising because we do know that E4 risk and dementia is actually strongest among Asian and white individuals and weaker in Black and Hispanic individuals. Okay. This shows the plot of continuous amyloid PET standardization uptake value ratios, or SUVRs, compared to percent African ancestry in the non-Hispanic Black group. So within the non-Hispanic Black group, those with higher African ancestry was associated with less amyloid, and this effect was independent of the number of ApoE4 alleles. So really, what does this tell us? Reduced amyloid was observed in self-identified non-Hispanic Black participants who actually passed the initial screening for A4. And this was really the first time we saw this. It was pretty surprising. We did not expect to see lower levels of amyloid based on race. And this effect has now been seen in a number of other studies, but A4 was really the first to demonstrate, and of course that did have an effect on eligibility. So we really need to consider diversity on what in our biomarkers, because many of these were initially validated in predominantly white samples. So this has really led to an explosion now, I think, and you'll hear about some of this, of wanting to look more closely at diversity in terms of our biomarkers and how they perform. Okay, another recent paper looked at sex differences associated with tau PET and cognitive performance. This was in a totally non-Hispanic white cohort of the A4 study. So we know that PET has been shown, tau PET rather, has been shown to be a strong predictor of cognitive, I'm not sure what happened with the slides, but I'll keep going, has been shown to be a strong predictor of cognitive performance across the full AD spectrum, including preclinical and prodromal AD. And so this study looked at this association of tau PET by sex in A4. There were 343 cognitively unimpaired amyloid-positive individuals, 205 women, 138 men. And this sex-stratified association between flotaucipir PET in the metatemporal region and the cognitive outcomes, the preclinical Alzheimer's cognitive composite PAC and the computerized cognitive composite C3. Okay. Well, if the next slide would come up, I would show you the results, but basically what was found was higher level of tau was significantly associated with worse cognitive performance only in women. And this was true on all the component measures except for one in the PAC, which was MMSE, and then all but one of the cognitive associations were APOE4 independent. So taken together, these results show that sex differences in the association between tau pathology and cognition in early AD, and that women with higher amounts of tau have poorer cognitive performance that you don't see in men. These findings have important implications for sex-specific tau targeted preventative AD clinical trials, especially when tau is an outcome. Okay, great. So that's what I just showed you, and that's just one of the scores. That's the overall PAC score. Okay. And then my last one, so I'm running really short on time now, is looking at subjective cognitive decline across ethno-racial groups. So they investigated this association between subjective cognitive decline and objective measures of cognition, as well as amyloid burden across diverse participants. There were over 5,000 non-Hispanic white, 262 non-Hispanic black, 179 Hispanic white, and 225 Asian participants who also did the PAC and the C3, as well as something called the Cognitive Function Index, which is the measure of subjective cognitive decline. And they also looked at measures of anxiety and depression. Okay, so the findings here were that the association between subjective cognitive decline and objective cognitive measures in amyloid were moderated by race. Lower cognitive performance was significantly associated with higher subjective cognitive decline in non-Hispanic white and Asian groups, but not in Hispanic white and non-Hispanic black groups. Same was true for amyloid positivity. The association was greater in non-Hispanic white and Asian groups and not in non-Hispanic black and Hispanic white. However, depression and anxiety were predictors of subjective cognitive decline in non-Hispanic black and Hispanic white groups. So this says that subjective cognitive decline reporting may not be as reflective of cognitive functioning in diverse groups, and therefore may be differentially predictive of AD pathological change, which is really consistent with some observational studies showing measures of psychological well-being are more strongly associated with subjective cognitive decline in these groups. So this is also an important feature to understand as we are looking at individuals to put into our trials how these measures perform. So, and I want to end on a really positive note. So the RCT data, the placebo and treatment arm data, will be available for A4 as of June 28. So stay tuned. We're really looking forward to that because the screening data has been very impactful. And this additional data, even though the trial was negative, is going to be highly valuable for understanding disease progression. So thank you all.
Thank you, Laurie. Welcoming Anna Greenwood.
Great. So I just learned recently that these are called confidence monitors, but since they're not working, I'm going to rename them as anti-confidence monitors, perhaps. But let's see here. Okay, great. So I'm going to tell you today about the Data Coordination Center for the Accelerated Medicines Partnership in AD that is led by Sage Bionetworks. And Sage, sort of similar to the FNIH, sees itself as an honest broker. We're a nonprofit organization that works with many communities to build team science expertise and share data across industry boundaries. And so the overall goals of the Accelerating Medicines Partnership, as it was first started, and I'm focusing here on the target discovery arm of AMP-80 1.0, was to shorten the time between the discovery of potential drug targets and the development of new drugs for Alzheimer's disease treatment and prevention. And really the way that this was going to be accomplished was integrating analyses of large-scale molecular data from human brain and fluid samples with network modeling approaches and really cutting-edge network biology, systems biology approaches, and then following through with experimental validation of these novel targets. And then a cornerstone and a key pillar of the AMP-80 program from its inception was really the open and rapid distribution of all the outputs of the program to the broader research community. And you'll hear much more from a few of the individual research teams today, but very broadly, the overall approach to target discovery that the AMP-80 1.0 teams took was to derive multi-omics data from multiple human cohorts and to use novel systems biology approaches to identify key drivers of disease, and then to use this data to identify potential new targets and to share these targets with the broader community and then do subsequent experimental validation studies. Another thing that the consortium did was to do a cross-consortium harmonized analysis. So the teams were each generating data often from different brain banks in very different ways. And there was a coordinated effort to bring this data together and harmonize it as much as possible. And then use this harmonized data set to do a follow-on set of analyses, including transcriptome-wide analysis, variant calls, expression QTL, and kind of a consensus meta-network analysis. And so this is a kind of by the numbers. So, and I'm focusing here specifically on multi-omic data, so data from individuals that are sampled across multiple data modalities. We have tens of thousands of patients, for example, profiled with just one single modality, but we have, here you can see 1744 individuals as of yesterday with not only bulk transcriptomics, but proteomics and whole genome sequence data. And then a subset of those also have metabolomics data as well. And the richness of this is growing as we add samples from diverse cohorts, which you heard was a major focus of AMP-80 2.0 as well. So 10 years ago, the AMP-80 was the sole source of data coming into what is now the 80 Knowledge Portal. And this infrastructure has allowed us to now incorporate additional 11 other NIA-sponsored programs into this network, and so to bring complementary data sets to share alongside of AMP-80. And so at Sage, we manage the data contribution from all of these programs, which represent over 100 grants now. And we bring the data in, organize it, and annotate it with a controlled metadata vocabulary in the Synapse platform. All of the governance is done by a team at Sage Bionetworks, and that allows users to request access, and we have developed a suite of external platforms to help with the discoverability and access to the data. So this includes the 80 Knowledge Portal, which is a platform for discovering which data is of interest, and then also several results explorers, Agora, which I'll touch on, and the 80 Atlas, which you'll hear about from my colleague, Matthias Arnold. And let's see. So as I mentioned, it's now 12 different programs, and even within AMP-80, the types of data that were being generated were extremely diverse and rich. So this is not only human samples, but the experimental validation arms also generated data from animal model systems, human induced pluripotent stem cell models, and even fruit flies. And all of this data, from raw data all the way to processed analytical results, is distributed widely to the broader community. This now encompasses over 60 different assays and 50 different file formats, just to give you a sense of the type of and richness of data in the portal. One of the ways that we sought out to help with the open science aspect of AMP-AD was kind of a multi-pronged effort here. So one is a commitment from the beginning, before our FAIR principles for findable, accessible, interoperable, and reusable data principles was even coined as a term, we were sort of embracing these principles. We also worked very hard to, and a lot of this was led by Susanna Petencheska, streamline data governance. So even before the genomic data sharing policy was initiated, we worked very closely with the NIH and other regulatory bodies to ensure that there was streamlined data governance, but protecting patient anonymity as much as possible. And so this was a really complex set of discussions that kind of goes on in the background, but the end point for users is that all data is available for general research use under one single data access request process. All of the data deposition happens just after quality control. There is no publication embargo. And another key pillar is that we don't just put the data out there, we work really hard to inform the community and educate them how to use the data. And so, as I mentioned, these FAIR principles were, our Synapse platform is compliant with all of them, and we continue to try to stay at the cutting edge of sharing data. Streamlined data access requests, we have different access requirements by data tier, and there's a very streamlined process for requesting access to that data. And then through our multiple support channels, which include documentation and a help site, an active discussion forum where people ask many questions about ROS-map data all the time, and we, and other data. We help them, and they learn. Users often learn from each other, and sometimes even answer each other's questions in the discussion forum. And then we have hands-on workshops, and really try to build a community around the use of the data. And we have tracked the use of the data and see that we have an increasing breadth of users and over 2,800 research teams, and sometimes teams can represent even tens, dozens of users within a single institution, have downloaded data between 2019 and 2023. You can see that academics are a major user of the data, but also non-academics, including pharma, biotech, government, and nonprofit users have leveraged this data as well. And one thing that we have really tracked is the secondary use of the data. So we know that consortium users are generating the data, and since they are the ones who are closest to the data, they're often, of course, publishing on that data as well. But a real mark of success for us is that most of the publications using the data are from non-consortium users, so secondary data use. People outside of the consortium who were not involved with the data generation are finding this data and leveraging it for their own studies. And so that just shows the power of open science, and it's going to take a collective team beyond just the consortia to really solve these complex diseases. I'm going to go ahead and, so we've had this platform up for about 10 years, but we continue to refine it. Some of the key areas we're focusing on include interoperability with other repositories. We recently added connectivity to a cloud compute platform called Cavatica, which also then the data can be accessed with many other platforms that are also compatible with Cavatica. Very quickly, since I'm running out of time here, I'm going to touch on our Data Explorer, which is called, our Results Explorer, which is called Agora. You heard this referred to by Dr. Collins as the wall of targets. And so this is originally designed to host a list of all of the nominated targets coming out of these discovery programs. And now it has grown from just a list of targets, but a compendium of molecular evidence coming out of these studies. And there are two main ways to access data in Agora, and it's really designed to not only aggregate the information about the nominations, the target nominations, but also to be used by non-computational scientists, bench scientists, experimental biologists, clinicians, patient advocates, patients, anyone who wants to should be able to use this platform. You don't have to be able to start from raw data and reanalyze it. So you can compare either molecular evidence for a single gene, like is my particular gene of interest associated with Alzheimer's disease in actual human brain samples, and you can also compare across genes as well. There's a real richness of data and resources available in Agora, and we specifically link to other great resources that have been developed by the NIH and broader research ecosystem as well. And with that, since I'm a little bit over, I'll just go ahead and end and thank all of the consortia and centers that have been contributing data to the Knowledge Portal, who we get to work with every day. And I just wanted to briefly highlight two members of the SAGE team, Jess Britton and Milan Vu, have posters this afternoon. So, thank you.
Thank you so much, Anna. And David Bennett is up next.
I just want to thank Susanna and the NIH and the FNIH for the opportunity to share some of our data today, which I may just be talking about, and you may not see it. Oh, I do? I have control. It's so unusual, because I came from my house where my wife has all the control. Okay, so I am going to be talking about disease subtypes in Alzheimer's disease. So first I want to acknowledge our funders, the NIH, the state of Illinois. I particularly want to thank the FNIH and the industry partners, the remarkable study participants in the 5 cohort studies that we run out of the Rush Alzheimer's Disease Center, where there's over 5,000 people that we've been following now for over 3 decades, almost all of whom are brain donors. And I have no relevant disclosures. So like a lot of the diseases that are in AMP, we're looking at complex diseases, and we're trying to understand how people and cells and organs move from healthy states to disease states, all right, and the interactions between the environment and the genetics that produce this. And then eventually, of course, we want to move disease states back to healthy states, and we'd like to prevent healthy states from ever becoming disease states. And so through AMP-AD 1 and 2, we've used our 5 cohort studies, the religious order study, which is a national study, everyone's an organ donor, the Rush Memory and Aging Project, which is Cook and the Collar counties, everyone's an organ donor, and then 2 African-American cohorts and a Latino cohort where organ donation is optional. There's rich antemortem phenotypic data. There's brain, blood, and other biomarker data. There's over 5,000 participants, and the studies are run by the same team where the clinical and the pathological data are harmonized, and we've done over 2,200 brain autopsies to date, and so far we've generated 110 iPSC lines, all from the same groups. Okay, so I'm a neurologist, and on the bottom you can see these syndromic phenotypes. So other clinicians in the room, we take care of patients, we want to make them healthy, so I can be a patient advocate. But our cohort studies are older people that don't have disease, and older people don't want to get disease. I don't know anyone that would like to get Alzheimer's disease and get treated. Most people would prefer not to get it at all. And so that's our primary focus. And then we are trying to get risk factors for disease and then understand how those risk factors cascade through different molecular events, all right, and then work their way into biomarkers and eventually syndromes. And so we have multiple layers of brain omics and blood omics, and we have multiple phenotypes in these individuals. So these are the most deeply phenotyped individuals on the planet. I think we have data for every single AMP with the exception of AMP schizophrenia. And so the motivating question for this particular talk is subtypes. Subtypes has been something of interest in Alzheimer's disease for a half a century. And the way we've approached it has evolved. First, there were clinical phenotypes based on clinical syndromes, which we've seen a lot in the history of neurology. And then we moved to neuropathologic phenotypes and subtypes. At least we're getting closer to what was the output of the brain, which was cognition. But now we're in the era of molecular phenotypes. And so through AMP-AD and various other projects, we generated all of this omic data. Oh, I'm in charge. Maybe I'm in charge, maybe not. Here we go. And so now we're working on putting these omics together to understand molecular progression. And so here on the left, and we'll ignore the multimodal data on top, you're going to see that we brought together 4 layers of omic data from the brain: epigenomics, transcriptomics, proteomics, and metabolomics. And then we can bring these together and create pseudotime trajectories. Pseudotime, in this case, is the molecular distance from normality to Alzheimer's dementia. Okay, how far are you away from normality towards Alzheimer's dementia in molecular space? And we can look at that molecular space in 3-dimensional space. And actually multidimensional, but it turns out to be 3-dimensional. And so you see 3 subtype trajectories, AD 1, 2, and 3, that are moving out in different directions. The distance is pseudotime, and the direction is the subtype. And so here's the differences between these 3 trajectories. AD subtype 1 is primarily driven by metabolites, although there's a fair contribution of CPGs and transcripts. Subtype 2 is primarily transcripts and CPGs with very little proteins and metabolites. And subtype 3 is primarily CPGs and transcripts, modest amount of metabolites, and no proteins. Now, I'm a neurologist. I like to take care of patients, and I'd like to prevent older people from getting Alzheimer's dementia, and here we are with all this brain data. So the next challenge that we're going to have in the field is taking all of this data that we've generated in all of these cohorts across AMP-AD, all right, and try and project it back to living people. And so the first thing we did was we looked at monocyte RNA-seq from a subset of the participants on whom we have the autopsies. And so we find 110 differentially expressed blood RNA monocytes, transcripts that are representing all 3 subtypes that we see in the brain. Now we're moving to augment these models with additional omic data. So here we've just completed creating subtypes where we add glycoproteomics, and we actually put in the neuropathologic traits. We get a new pseudotime, slightly different. We still have 3 subtypes. That seems to be fairly consistent no matter how many layers of omics we put in. Okay. Okay, but now we've really augmented the blood data. So now we have cell-free DNA. We have the monocyte RNA-seq from before. We have proteomics, metabolomics, and I also threw in p-tau 217 and 181. And what you see in the middle on the bottom is the cross-validated AUCs. And on the bottom row, you'll see the p-taus. Now, the p-tau ROCs for predicting the 3 subtypes, okay, is basically chance. So the most common thing that we're measuring in the blood as an AD biomarker, and I'm not saying it's not predicting Alzheimer's disease or tau in the brain, or what I'm saying is it's not predicting these subtrajectories. But you're getting AUCs of 0.7 to 0.8 across the different layers of data with the exception of cell-free DNA. So we're beginning to really move from the brain to data we can get from a living person. So we initiated integrated epigenomic, transcriptomic, proteomic, glycoproteomic, metabolomic, neuropathologic data. We generate a pseudotime. We generate 3 subtrajectories. We can translate these subtrajectories into blood omic data. And then the ongoing work is to both augment the models with additional layers of brain data. I just got another one this morning. And then to bring in other layers of data that we have from the same people from living, which is structural and functional MRI, other layers of blood omic data, and eventually the PRS. And the next steps is to move to experiments with these subtrajectories. And that would be in iPSC lines, all right? And the first step there is going to be to try and see if we can generate these subtrajectories with multiple omic data from the iPSC lines. And I'm looking at you, Tracy. And so I don't have time to show you that data, but Sarah and Tracy will have a poster this afternoon, and I encourage you to go look at that. So I want to thank you all for your attention. I'm pretty sure that's my last slide. And I'm going to turn it over to the next speaker.
Thank you, David. And we're moving on to integrative proteomics with Nick Seifert.
Great. I want to thank the organizers, Susanna and the FNIH, again for giving us an opportunity to talk about our work and our progress over the last 10 years. So I thought I'd share sort of the progress in terms of the number of tissues that we've analyzed by mass spec proteomics since the inception of AMP-AD. And actually, it was funny looking back at the first round of when we wrote this grant, we actually thought it would be a really big achievement if we hit 500 brains. So now we've had 5,000 brains, and we've done this both by deep quantitative proteomic analysis, which generates about 10,000 proteins per an individual brain sample, and also targeted analysis, so we're able to actually really drill down on those key drivers that we talked about earlier to measure these across a number of samples. We've done it in 12 independent ADRC brain banks, and importantly, we've done it in over 250 African Americans and 300 Latino Americans, and those data are forthcoming in a scientific descriptor. We've also done this in biofluids, both cerebrospinal fluid and plasma, and we've partnered with ADNI to do all baseline ADNI CSF samples. That's about 1,200 to 1,400 baseline samples. And then the autosomal dominant AD, Diane, in collaboration with Randy Bateman, we've done 1,000 of those longitudinally, and that's given us a better picture of how these biomarkers change over time with the course or the evolution of disease. So here's the high-level overview of what we do with this data, and I think the biggest challenge with AMP with data is how do you make data into knowledge? How do we translate this into something actionable, not just from a therapeutic target point of view, but also from a biomarker point of view? So we do this by using network-based approaches. So we can take these complex data sets and reduce them into communities or modules of highly co-expressed proteins. And then once we have these, we can leverage other orthogonal data sets that are in the literature, let's say single-cell omic data, and define whether these communities reflect, let's say, synaptic biology or endothelial biology or neuroinflammation. We can then take these modules, which we sort of consider the biochemical endophenotypes of Alzheimer's disease brain tissue, and we can look at them, how they change across the stages and the transitions of Alzheimer's disease. Do the proteins in these modules occur early, prior to cognitive decline? And do these proteins change late? Would be more reflective of something that correlates with symptoms and symptom onset. And then what we do is that we've also been able to integrate this with genomics. And one of the real pleasures is that we have paired either genotyping or whole genome sequencing in a lot of these. And this allows us to do something with functional genomics and assign pQTLs and then co-localize these QTLs with GWAS to see if these are causal targets. And this helps in terms of our prioritization or go, no-go decisions. But I'm going to talk in the next few slides on how we've been able to translate these markers from the brain into cerebrospinal fluid markers. And so one goal we wanted to do in the AMP-AD was to say, if these proteins are changing in postmortem brain, are they reflected in peripheral biofluids? So we've taken these co-expression networks, we build modules, we assign them to their ontologies and their cell types, And at the same time, we did deep discovery analysis of cerebral spinal fluid from patients with Alzheimer's disease. And we then overlapped, we integrated, and seen if we could see those same changes that happen and relate to pathology in the brain, are they manifesting in the spinal fluid? And what was surprising to us is about 70% of the proteins that we could detect in the cerebral spinal fluid did in fact have an overlapping protein in the brain. So it did support this hypothesis that the CSF was this window into the brain. So the next thing we wanted to do was further collapse this data, because you can get several, let's say 40 to 50 modules in a network, but we wanted to really collapse them into actionable biomarker panels. And we thought there would be kind of 5 main ontologies that we wanted to capture, one of which was synaptic or neuronal biology, one was vascular or endothelial biology, myelination in terms of white matter and oligodendrocyte biology, glia, microglia, astrocytic and inflammation, and also metabolism. And metabolism is very correlated to the onset of cognitive decline. And then we were able to do is that once we had this integrative pipeline, we could use a data-driven approach to nominate peptides that were reflective of these brain proteins in the spinal fluid to come up with targeted panels that we could run at scale. So instead of this laborious work of doing this discovery, we can now run a panel of, say, 60 to 70 proteins in 15 minutes and run 100 samples per day. And then what allowed us to do is then do all of ADNI, for example, and then see if these panels were better than the standard amyloid and tau and phospho-tau in predicting cognitive decline from baseline. And in fact, they were. It was a paper published by Rafi Haig last year, Science Translational Medicine. And the other interesting, using longitudinal data, we found proteins such as SMOC1 that were actually accumulating in the cerebral spinal fluid upwards to 30 years in advance of cognitive decline. This is actually coincides with the amyloid deposition in the brain by PET imaging, to tell you that there are other biomarkers that are in the CSF that are reflective of those underlying pathophysiologies in the brain. I'm going to click data sets such as those from AMP-PD to look at the specificity. And we're seeing here, as I talk about this protein SMOC1, which was up 30 years in advance of cognitive decline in the DIAN cohort, it is definitely increasing specific to Alzheimer's disease. And if you do neuropathological assessment of proteins like SMOC1, they're very specific to amyloid deposits within the brain. They bind parenchymal plaques, and they also bind CAA, the vascular amyloid in the brain. And I think this is a nice example of leveraging other AMP-AD data sets to get at the specificity of these changes, because we want things that are specific potentially to Alzheimer's pathology that could classify someone with Alzheimer's disease compared to other neurodegenerative diseases. And another effort we have, and this is work that was done by a graduate student in the lab, Erica Modeste, was to compare these brain-based CSF biomarkers across race. And we know that from studies from John Morris and William Hu at Emory and John Morris at WashU, that African-American CSF levels of tau in African-Americans are significantly lower than Caucasians with Alzheimer's disease. So this has profound consequences in terms of enrollment into clinical trials. And what we see here is this is over 203 CSF samples in which half of the participants were self-declared African-Americans. On the left, you have the Roche Alexis immunoassay for tau, and on the right you have the mass spectrometry measurement for tau. And you can see in both assays we see a significant reduction or lower levels of tau in the CSF of African Americans with AD compared to Caucasians with AD. But the question remained, was that the only thing that was different between the races? And what Erica found is that using these targeted panels is that a lot of these synaptic markers that correlate really strongly with cognition in life, such as VGF and neuropentraxin 2, are also lower in African Americans than Caucasians with Alzheimer's disease. And this kind of conundrum, because contrary to predictions based on CSF tau levels, African Americans had lower levels of these neuronal markers, suggested that they may be more on the ATN continuum that you wouldn't normally predict from tau alone. So it's a good insight, and now we have unbiased CSF from a much deeper cohort now that we're going to be able to leverage and then map back to see if these changes are actually happening in the postmortem brain samples. And one of my last slides here is, we can correlate, we can associate, but one of the things about biomarkers is that if you engage it with a drug, pharmacologically, do those biomarkers change in response to treatment? And this is the idea that these proteins could be theragnostics, and therefore that could actually start to pinpoint mechanisms of action of a potential therapy. So what we were able to do, when the slide comes back, here it is. What we're able to do here is that we ran a clinical trial at Emory, led by Alan Levy and Jim Law, and where we repurposed a drug called Strattera, which is a norepinephrine reuptake inhibitor. It's given for ADHD, and it had some influence in animal models in reducing the level of tau. And so we ran this clinical trial, and then we took the baseline and then the post-treatment, both the placebo and the treatment arms, and you could run these biomarker panels to see how these underlying brain-based biomarkers, or CSF proteins, were changing in response to Strattera treatment. And it was interesting, we would have thought going in that it was going to be neuroinflammation, because most of the studies had shown that it would be reduction of glia activity and reduction of inflammation. But the two panels that showed response to Strattera treatment were the synaptic panel and the metabolic panel. And it was interesting, I think some work by Oscar Hansen now is showing that these metabolism markers are very correlated to tau PET in vivo in their biofinder studies. So this is sort of a reflection potentially of engagement, indirectly engaging tau in the brain. Okay, so where do we go from now? We've heard a lot about CSF subtyping and molecular subtyping. David did a great example of that for the brain. Betty Times and Visser had a paper earlier a couple of months ago about CSF subtypes. We were very proud to see that they used our AMP-AD data to validate these subtypes. And Maddie Bangs will have a poster on the subtyping we did in the AMP-AD, and I can say that they're very consistent to what Tim's and colleagues have reported, which is a good thing for the community. But as we want to know more, we want to learn about whether these subtypes are related to comorbidities, whether these subtypes reflect different degrees of progression, whether the subtypes respond better or worse to specific treatments. I think that's going to be important. If there's going to be a specific drug, can we actually target a specific class or type of AD? And which patients have higher risk of adverse side effects? You know, with the ARIA that's out there, we would like to know if anyone was actually at risk before you enroll someone into those trials, or actually for those treatments at this point. So with that, I'll wrap up. It takes a village to do this. I've been very well supported by the group and by mentors throughout the AMP-AD program, and of course our funding from the FNIH and the NIH. So thank you very much.
And our last research vignette by Dr. Matthias Arndt. Thank you, Nick.
Okay. All right. Thank you very much for the introduction. Thanks to the organizers for giving me the opportunity to present our work here. So I'm standing here as a representative of the Alzheimer's Disease Metabolomics Consortium, and while I will be talking about multi-omics integration today, I just wanted to quickly show this slide, which shows the data that we have generated within AMP-AD, which is covering 60,000 profiles of human samples from blood, brain, and CSF across different cohorts, and also covers now almost 2,000 samples from the newly generated mouse models of Alzheimer's disease. However, for one of the central goals of AMP-AD, which is the identification and prioritization of novel targets for therapeutic intervention, we actually didn't want to focus on only one omics type, but what we wanted to do was to embed single omics disease associations into the wider context of multi-level molecular changes. And on the way, we also wanted to address common challenges in integrative research, which are data heterogeneity, access restrictions, and sometimes missing bioinformatics resources. So here is a summary of what we did. I will go into this in a bit more detail in the following, but what we did was we first started to collect large-scale multi-omics data. We then processed and harmonized the data, so not so much for AMP-AD, but for external data, because AMP-AD had its internal harmonization pipelines. We then developed an integrated data model to represent all of these different data types, right? And we were going actually to build molecular networks. So we built a model for an integrated multi-omics network model. We also wanted to prepare a web interface to enable public access to the data that we generated, and then also developed some tools to leverage the collected evidence that is available in the platform. So building multi-omics networks is rather complex. Usually what you assume is that you will have to have all of the different omics data types within the same cohort, which is rarely the case, right? But there are also alternative approaches, something that is called stepwise data integration, where you use data from different cohorts and leverage them to the extent possible defined by the availability of the different omics data types. So imagine you have one cohort where you have just one type of omics data. You can still use that, right? You can use it to, for instance, calculate intra-omics correlations like gene co-expression networks. If you have, in addition, phenotypic data, then you can add for that omics layer associations with disease. In the best case, of course, you have several types of omics data, and then you can calculate all three types of associations that we have, so intra-omics links, phenotype-specific links, but then also inter-omic links, like, for instance, if you have GWAS data plus one type of molecular omics, like metabolomics, you can calculate genetic associations for that and thus link genes to metabolites, for instance. Those are the data sets that we have integrated so far. Very briefly, what we did is we started out with integrating data from population-based cohorts because we wanted to have a network structure that in general is not specific to a disease context. So it can be used basically for everything. And that we then turned into an Alzheimer's disease-specific resource by adding large-scale association data for Alzheimer's disease, both coming from large GWAS screens, but also from the multi-omics experiments that have been done, for instance, in the AMP-AD consortium. Now here you see the data model that we came up. Detailed view is given on the left-hand side, and as you see, this is quite complex, and we actually found that this is too complex for both good visualizations and also straightforward analyses. So we had to come up with an abstraction of that, which is shown on the right-hand side here. And the most crucial step there was actually that we compressed information from epigenetics, genetics, transcriptomics, and proteomics and summed them up into gene elements. So we basically collect all of those different types of evidence in one type of node. And then we also manually curated metabolite annotations across different platforms such that each metabolite is only occurring once, and also came up with a controlled vocabulary for Alzheimer's phenotypes. This is the content of what we are covering right now. So we have 43 different clinical pathological traits and biomarkers. We're annotating all protein coding genes in the human genome, many of which also have transcriptomics and proteomics evidence available. We have data on 1,300 metabolites, and all of those are interconnected by close to 1 million significant associations. Now, I said we wanted to enable public access to that data, so we built a web-based user interface that is available under adatlas.org. Here you see a screenshot, so also the web-based interface is all about networks, as you see here. The general idea, as I said in the beginning for the AD Atlas, not only the web interface, but also the AD Atlas itself, is basically augmenting single putative targets, not only across omics layers, but also across their functional neighborhood, because we assume that if a target really is promising, then it should also be reflected in its functional neighborhood, which we define as a one-step association network around it, as we have it in the AD Atlas. And this is here shown as an example for ApoE. We'll see if you start with one gene, get the functional neighborhood, you end up with a network that already covers more than 170 genes, and that can then be already used to add information through, for instance, a gene set or pathway enrichment analysis. Right, so I don't have too much time to go into more detail about the user interface itself. Just wanted to mention, so we have actually prepared several use cases on the website that show how it can be used from A to Z to extract relevant information from the database, covering both approaches towards drug repositioning, but also more exploratory analyses. But as I said, so we didn't only want to harmonize all of this data, make nice visualizations, and look at small-scale examples, but we actually wanted to use the entirety of the data for deriving novel insights. And I will go into two examples today. The first is unsupervised identification of disease modules. And what we did there is we first transformed the network using a deep learning-based approach that is called network representation learning, which basically takes the network and transforms it into a numeric matrix, which can be likened to a structurally weighted adjacency matrix that's also a bit compressing the complexity of the data. We did that because that allows to apply the common approaches for unsupervised clustering, for instance, and then also can be used to do nice visualizations of the whole content of the network. Here we also filtered this down to a brain-specific subnetwork, meaning that we only extracted molecules that we actually measure in brain, and we also removed all Alzheimer's disease information, because what we wanted to learn was actually just the multi-omic structure, and then do module identification and find then those modules that might be enriched for Alzheimer's disease relevant information. So the result of one of these approaches is shown here. So on the left-hand side, you see the result of a hierarchical clustering that we did based on this numerical matrix. There's one cluster highlighted. And on the right-hand side, you see actually 3 times the same picture of the 2D UMAP representation of the embedded network, colored by different information types. So at the top, we have functional enrichment, which here shows a significant enrichment for immune response for the highlighted cluster. We then also look back at the AD information, right? Here, seeing for this cluster a significant overrepresentation of genes that are overexpressed in the temporal cortex of AD cases compared to controls. And on the bottom, we had another network-based measure for disease relevance where we look at the average shortest path for all elements that are included in this cluster, which in this case showed that this immune cluster is more tightly linked to Alzheimer's disease than you would expect by chance. So basically this suggests that despite the different data sources that we used, right, and different technologies and so on, basically the integration approach and the harmonization actually works and let us retrieve AD relevant information from this global network. The second approach that I wanted to just briefly touch upon is computational drug repositioning. For that, this is part of another project that's called Target AD. Here we use the harmonized data within the Alzheimer's disease atlas by 2 ways, right? So first, we extract disease signatures across the different omics types, so transcriptomics, proteomics, and metabolomics, and then do signature-based drug repositioning predictions. On the other hand, we use the high-confidence network structure within the Alzheimer's disease atlas to actually employ network-based repositioning algorithms, and then we bring them together in a weighted rank aggregation method, which then allows us to use both types of information and retrieve the candidates that are actually top-ranked across all of these different methods. And then we can annotate them by, for instance, looking into how well they align across different brain regions or cell types. I just wanted to briefly mention that the ID Atlas is an evolving tool, so we are continuing to develop it. We also enrich it with new data continuously. Just a few example projects that add to that. AMP-AD, of course, we are still not done with integrating all of that data. We also add data from the AMP Systems Biology of Inflammation pilot. The Alzheimer's Gut Microbiome project is adding microbiome and exposome data, Target ID I just mentioned, and then the Alzheimer's Disease Metabolomics Consortium is also involved in additional initiatives which are mostly focusing on metabolomics and lipidomics analyses. With that, I would like to thank my partners in the Alzheimer's Disease Metabolomics Consortium. I also would like to thank our partners in AMP-AD for some really great collaborations, and would like to end with pointing you to 2 posters of our consortium that will be presented by Priyanka Baloni and Richard Batra in the afternoon. Thank you.
Thank you all for a fantastic set of presentations. And now inviting Rebecca Edelmeyer, our steering committee member, to share her reflections.
Thank you so much, Susanna, and I really want to thank the NIH, the FNIH, certainly Susanna, Lori, Mike, for inviting us to be able to share our perspective today. The association and the NIA have really been working together for decades now across AMP-AD 1.0, 2.0, and many, many other projects, and it's really just incredible to be able to see the progress that we are making working together. As the largest nonprofit funder globally of Alzheimer's and dementia research in the world, we really see absolute value in these types of public-private partnerships that allow us to work across sectors, really working together, that will help to create a drive collaborative science, really truly open science. And I think, as you saw demonstrated by Anna this morning, portals that allow scientists to really interrogate that data. It's not just science for science sake. And we're really continuing to advance on a scale that we would not otherwise be able to do, I think, if we were working in our silos. And I think that really demonstrates the importance of our work together. We're advancing research on a scale, I think, that has never been done before. And I think it's important to also note, though, that it's not just about the science. It is about the people, and I think that was brought up in our first panel discussion this morning, how critical it is that we keep people at the center of our work. For many of you in this room, I'm sure and I suspect that the work that you do is very deeply personal. It is for me. It is for many of the families that we serve through the Alzheimer's Association around the world. And I think the work that we continue to do as researchers needs to reflect all people. And I think what you have seen in the transition from AMP-AD 1.0 to 2.0 is really truly a deep commitment that the consortium is working towards in terms of diversifying the samples that we're studying. We must really continue to understand the contributions of the disease in populations that are most at risk for Alzheimer's and other dementia, especially individuals that are of African American descent, Hispanic or Latinx populations. There's really a need for rich phenotypic data that is representative of all individuals so that we can continue to develop discoveries that are going to be benefiting all people. We need to understand, I think, what that rich phenotypic data actually says. We need to understand the distinct molecular subtypes about who is affected and why, so we can continue our drug development. And we're really in an era of biomarkers and multi-omic systems analysis that can really help to support this work. There will be challenges, I think, as noted by Matthias just a moment ago, of integrating all of the multi-omic data together. But it's a challenge we must accept in support of development of these new treatments. We know that Alzheimer's is incredibly complex. It's likely that it will be most effectively treated through a very powerful combination of therapeutics that target different aspects of the disease biology, as well as even powerful risk reduction strategies for all individuals and all communities. And we're living now in an era of new treatments for Alzheimer's disease, so it's a truly exciting time to be doing research in this space. With the work that we're doing with AMP-AD, we know that it will help us to move towards a more person-centered and precision medicine type of approach for Alzheimer's that support a diversity of treatment options that are FDA approved, that will be available for patients across all stages of the disease continuum. And as an example of how we might continue to build a future in terms of drug discovery, we want to make sure that we continue to look at the data in a variety of ways, that we use this maturing data set that exists in the AMP-AD knowledge portal, and we're promoting opportunities for researchers to use that. Example of what the association has done in partnership with the NIA is that we continue to create funding opportunities. One would be the ALS Discovery Grant program that we just launched last year, where 10 researchers were actually funded to really use data that has been put into that AMP-AD knowledge portal, as well as Model AD consortium contributed data that helps to support preclinical animal model development for Alzheimer's. So that continues to move the needle forward in making sure that we continue to promote the use of this data and not just continuing to collect the data. We know that there's more work to do together. We must really definitely continue to advance the pipeline from discovery to clinical trials, all the way to this new horizon that we're now entering, which is the collection of real-world data that will help to support real-world evidence generation. And we need to continue to do that as partners together. These types of collaborations are really strong evidence. We truly believe that increased federal funding, increased funding from nonprofit organizations like the Alzheimer's Association, that their Alzheimer's and dementia research is being done and utilized in the best way possible and as fast as possible for the patients that we serve. So thank you very much for listening, and thank you very much for allowing us to be a part of this consortium.
Thank you, Rebecca, and we'll conclude with Mike's reflections.
All right.
I'll let the slides work. So I'm really pleased today to give a industry perspective on the value and impact of AMP-AD. And actually, before I set out to kind of come up with this, I reached out to a number of my industry colleagues on the steering committee because I recognize that I'm giving my perspective, but I also wanted to capture my other colleagues' perspectives. So just briefly, I wanted to touch on 5 different areas that I think bring real value to industry partners. First, we wouldn't be able to do any of this without the immense amount of data that's been generated from the consortia. So as several speakers have already talked about, the expansion of the multi-omic data has really kind of transformed how we viewed the disease pathophysiology, and especially really diving into these diverse cohorts to expand into these areas in order to better represent the disease and also the patient diversity that we see, not just in the general population, but also as we see it in the clinical trials that many of the industry partners and nonprofit government are trying to recruit for. One particular area that AMP-AD has really driven quite a lot of research into is expansion of really in-depth molecular phenotyping, in particular single nuclei profiling. This has really kind of transformed how we view the disease, and you've heard a little bit from Nick and others about the need for understanding different cell populations based on the data that we're seeing, and really this has allowed for deeper investigation of the disease biology and mechanisms and allowed us to kind of explore new target space. An area that AMP-AD has really kind of advanced into, and you've heard all 3 presenters as well as Anna's presentation on new methods and analytics. And so these methods are being developed by AMP-AD, is also being applied by AMP-AD investigators, as well as industry partners. And I know many of my partners would agree that we're really trying to utilize the data and also integrate it with other data sets to derive new novel targets and mechanisms. And we've already heard today from Matthias, Nick, and David about contrasted PCA and pseudotime analyses, representative network learning. So we're really trying to advance a lot of the analytic approaches that we're using and in many different contexts, but also in the context of AD. This work would not be fully realized in terms of new targets unless we actually explored in an in-depth way the nominated targets that are coming out of the computational approaches in complex model systems, including iPSC-derived neurons, organoids, other approaches, and this really allows for de-risking these targets. So you've heard from the panel earlier today that the targets themselves are not just enough. We need actually to investigate the biology once we've nominated these targets, and this really allows industry partners to lower the threshold to be able to introduce these projects internally. So it's really important that we actually have these platforms in the public sphere and work collaboratively with our colleagues from the academic groups to validate a number of these targets that we're looking at. In terms of infrastructure, you'll hear tomorrow about data everywhere, but there's a massive amount of data that's being generated, terabytes, almost petabytes probably worth of data that we're getting close to. And it wouldn't be possible without coordinated and centralized storage and access to data, results, and targets that are being generated by AMP-AD, by the folks at Sage, as well as the folks that are developing these different platforms to view the data, like the AD Knowledge or the AD Atlas. And so this really democratizes the insights that can be derived from this resource. And as was spoken about by Anna, the vast majority of access to this data is coming from people outside of the consortia, so this wouldn't be possible without the infrastructure that's in place. Finally, this was something that I believed in, but also I heard from a number of industry partners, was that the open sharing of ideas and data and close interactions with our academic colleagues and experts has really been enriching, and it allows industry partners to form better, more informed target identification and validation efforts. But it's just really scientifically enriching to discuss these things with our academic peers. So we're really grateful for the opportunity to interact with them on a regular basis. And with that, I'll close my remarks, and I think that's the end of our session. Thank you.